Good morning. We've heard in this conference quite a lot about how HiFi sequencing is helping us with the human health. And my aim today is to say something and convince you that HiFi sequencing also is helping us address many of the other challenges that we face globally: conservation of biodiversity, food security, energy security, and particularly our efforts to use it to address reducing climate change. So I'm going to talk across that range of applications. First of all, starting with conservation, I'm working with the Australian Institute of Botanical Science, where we're applying genomics to rainforest regeneration and conservation of endangered species. We're now using genomics to guide the selection of seeds for all plantings by farmers and others of restoration of environmental sites, and we're moving to whole genome sequencing, and we need, of course, the HiFi references to make that work for every species that we plant in the rainforest. This species here is an example of a recently described species. We had 90 individuals of the species. We now have a high-quality HiFi reference genome, but there are only 45 individuals left after a recent fire. So in critically endangered species, we aim to have a genome sequence for every individual, not every species. And that's an important way of critically managing endangered resources. But what we're about is capturing that biodiversity to support food security and energy security, and this is a list of the major food crops in which we're using HiFi sequencing to characterize the genetic diversity that's available for incorporation into plant breeding programs. And of course, we all know about the advances in DNA sequencing, and we've been on the very rapid path in the last few years, since 2020, with the advent of HiFi sequencing and the rapid advances in our ability to de novo sequence genomes and to discover a whole lot about the biology of the plants and how we can utilize them in our crop breeding programs. To some extent, the culmination of that for us has been our recent sequencing of the mango genome. Here we were able to get all chromosomes as a single contig with telomeres on both ends from HiFi sequences alone. No other information, no genetic maps, no HiC, nothing else. And I think increasingly HiFi will give us a complete telomere-to-telomere genome with no other information. We don't even need to know the number of chromosomes. We determine that by the number of contigs that we get when we HiFi sequence. And this is proving true for even species with very large numbers of chromosomes. So quite a milestone, I think, that we've got to that point that the accuracy of the sequencing allows us to do that. A further illustration of that is our recent sequencing of the rice genome. Now, you'll say that that was done before. Rice was the second plant genome sequenced in the early 2000s, and I wrote a review of progress over the last 20 years to mark the 20th milestone, 20th anniversary of the sequencing of the rice genome. Now, we sequenced it with HiFi, and we found that there's about an extra 10 megabases that was missing in the reference genome that people have been using. And there was a few hundred genes there that have been missing. But much more importantly, in the regions we already knew about, across the whole genome, there were many more genes we could now annotate because of the greater accuracy of the HiFi generated genome. And we've been able to find at least 10% more genes in the rice genome by HiFi sequencing than in the reference genome that the world has sort of focused on. That was a result of, in fact, many thousands of person years of sequencing and annotation for our teams in many countries. So it shows how the technology has overwhelmed our past resources, and the fact that we actually need to revisit all those major genomes that we work with and resequence them with HiFi to get the accuracy we need to unlock many of the genes that are missing in those genomes. I wanted to say a little bit about polyploid genomes, because that's where HiFi really has helped us a lot. A lot of plants are polyploid, and that's a big challenge. It's been an impossible challenge for many species. Coffee is a good example. We've published the coffee genome, and that was published in Science back in 2014, the Robusta coffee genome. It's a diploid, and that was relatively straightforward. But the tetraploid, we only published this year in Nature Genetics, with the availability of HiFi, because we could get the tetraploid genome to the level that we needed for publication. And it took 10 years longer to do the tetraploid than the diploid. That's an illustration of the relative difficulty, I guess. But sugarcane has been the ultimate goal, and we've worked towards the sugarcane genome sequencing for more than a decade and thrown every technology that's available at sequencing it, and been frustrated by the fact that this genome, because of its great complexity, has been really challenging. But we've just, a month or so ago, managed to publish this genome in Nature. And the reason we could publish it in Nature, it's probably the last plant genome that will be published in Nature, and that's because it is the most complex of the 20 major crops in the world. And it's the last of the 20 major crops to have a genome because of that complexity. But we were able to sequence it, and we'd almost given up a couple of years ago after having spent really too much money on this. Everyone in the world, JGI had given up because they didn't have the resources. But I sequenced a few HiFi cells and sent the data to JGI, and that revived the whole thing because suddenly the assemblies worked and we were able to put together this very complex genome. We've got other examples. So we've just discovered a species in the desert that's also another dodecaploid, 12 copies of every chromosome, and we've got them all with HiFi. This is potentially a new crop that we could domesticate that's adapted to desert environments. But climate change is the big driver of plant breeding today, and this is just showing how the temperature's changing, and we need to address that with new cereals, with new pulses, with new versions of all our major crops that are adapted to those the climates of the future. And so we're systematically working through all of the flowering plants. I think I've looked at every plant family now in terms of what might be in that family that could be eaten or could be developed into a crop. And so we really need to look at the genomes to really understand the potential of these species. So we look at crop wild relatives, and sometimes species where there are no relatives that were currently domesticated, but that might have potential if we worked on them. And so, but the big focus is the wild relatives of crops, and quite often we go back to think about where we came from. Barley, this was the first plant that was domesticated, probably 12,000 years ago. We've moved on from that to thinking that really the overlooked area is where we are now, the Asia Pacific. So we've got a strong focus now on what happened in crop domestication here. Traditionally, I think we've had a Eurocentric view of crop domestication from the Mediterranean and those areas, which were important areas for the beginnings of agriculture, but in fact, we've overlooked the fact that at the same time, there was a lot of crop domestication here. And if we think about it, where we are now is in the center of this circle where half the world's population lives, and this is where the food's needed, and this is where agriculture is actually developed greatly and needs to be better understood in terms of not only the past activities in crop domestication, but the current and future potential to source biological material from this region to support global food security. And in Australia, we're going back to work with Indigenous peoples. We've had food production for more than 60,000 years in some of the desert areas, and we're trying to discover the plants that were used for food all that time ago because we lost the knowledge on that. And so we're working with archaeologists to identify in the contemporary flora which of those species have potential for future domestication. So looking at crop wild relatives, we're looking at coffee. We've got an international effort now to sequence all of the coffee wild relatives. Coffee is threatened by climate change because the areas where we produce high quality coffee, high altitudes in the tropics, and those climates are being lost. So we're less likely to be able to produce premium coffee without new species. New cereals, of course, we need calories. We're working on sorghum. Sorghum was domesticated in Africa, but we're now with JGI sequencing the entire genus with HiFi to allow us to get a pan-genus genome that will allow us to drive what is probably the best adapted cereal for warmer climates into the future. Many species, all of which are found only in Australia. Rice, this is the local region that we work in with rice, from northern Australia to northern China and across to India. This is the local patch, and across that region we have great diversity of wild rices with poor genomics in most cases. We have high quality genomes for the Australian material, but less high quality for much of the material throughout Asia, and we need to collect and sequence that to really understand the diversity we've got to work with in trying to face the future with rice. We've done detailed work on about 7 species in Australia and beginning to understand the origins of rice. Rice has been domesticated in Africa and in Asia, but there's a great gene pool there that we can work with. And in Australia, what we've found is that we're seeing natural hybridization between the species in the wild, so that rice is a product of reticulate evolution, and so we've discovered that many of these species have actually contributed to domesticated rice. It wasn't a simple story that rice was domesticated from a single species. We now see that there was integration from maybe 4 or 5 species into the gene pool that contributed to domestication. And that story is just beginning to emerge, largely driven by the fact we have HiFi genome sequencing to be able to de novo sequence all of the wild material. So we're really aiming to reinvent rice. We've got rice now with bigger seeds in the wild than we've got domesticated, an obvious opportunity for improving rice. We've got all sorts of interesting properties, including slower digestibility, which will be important in human diets, that we can bring from the wild into the domesticated gene pool. And we're using genome editing. So we need a highly accurate genome to edit it, and then we need to have things like HiFi sequencing to be able to sequence all the products of editing so that we can get regulatory approval to release them into the environment to show that we've edited what we thought we'd edit, so we can rapidly deploy into agriculture gene-edited, wild domesticated material. So we're domesticating a number of new crops using gene editing, relying on the fact we've got accurate genome sequences to edit and that we can analyze what we produce. This species will represent a new type of rice. It's the most distant of the close relatives of rice in the primary gene pool, the AA genome pool, and that looks like it's going to be relatively easy to domesticate. We're also domesticating another rice species that has novel starch properties and that survives the dry season as a rhizome, a perennial rice that looks like it's domesticatable. Citrus is an important area for where the HiFi sequencing has really helped. The world is facing a major crisis with orange and citrus being attacked by an HLB disease that has pushed up the price of orange juice globally because of decimation of commercial citrus crops. We're sequencing a whole lot of wild species, including discovering new species from HiFi sequencing. So sometimes we can get ahead of the taxonomists. We can sequence something and realize it's actually a new species. That's exciting when we can do that. And here we've found, with high quality sequences of the wild material, that we can identify disease resistance genes that we can use to deploy globally to defeat this disease. We've got a number of species that are fully resistant to the diseases that are currently attacking citrus, and so we can use this knowledge of the disease resistance genes to back cross disease resistance into commercial citrus, and that's happening at a great rate, all driven by the availability now of the genome sequences. We've done the same with the wild macadamia, and the big discovery for us there was that there was a whole genome duplication event that meant that nearly 30% of the genes are duplicated on a second chromosome, and that's really helped us understand the breeding of this relatively newly domesticated species. Banana is the same. Wild bananas are a source of resistance to the fungal diseases that are threatening global production at the moment. We have a new genus of grapes that we've discovered from DNA sequencing. We've identified that it's actually not just a different species, but it actually represents a different genus from the analysis of the diversity we're seeing. And the big opportunity, I think, that remains untapped is new pulses. The world needs more proteins. We've got a lot of wild species that haven't been domesticated. Their genomes will guide us to rapidly do that, to diversify the range of protein crops that we can grow. We're working with some remarkable legumes that grow in the desert that seem to be very productive, just the sort of things we need for climate-adapted agriculture. Pigeon pea is a great example where we're bringing insect resistance from the wild into the domesticated gene pool. I'll just say a little bit about horticultural crops. All of these species we now have HiFi genomes for, and in fact in many cases for very many species within each of these groups. We've completed the top shelf there, the mango, macadamia, avocado and citrus. We're now working through the bottom. Pineapple and banana have been particularly successful. The banana genome we have great results for all 33 chromosomes. This is a triploid, which really defied genome sequencing with previous technologies. But bananas are a very important crop, and the HiFi sequences really make a big difference to this crop. With pineapple, we have the first haplotype resolved pineapple genome, and it shows that pineapple is incredibly heterozygous. Really, the collapsed genomes we've had before are fairly useless because the two haplotypes are just so different. You need to know about both of the haplotypes, and that has been relatively easy, of course, with HiFi sequencing. Another, I think, interesting example of application is to dioecious plants. This is where you've got a separate male and female plant, and so we've sequenced the jojoba genome, and what we discovered, the first time anyone had de novo sequenced a dioecious plant, remarkably, that the male genome was much bigger. The Y chromosome was 10 megabases longer than the X, and really people had just played around with markers in the past. No one had really de novo sequenced a dioecious plant, and this allows us to understand sexual dimorphism in plants, and it's quite extreme, much more so than in humans where the difference is almost trivial. In plants we see massive differences and separate evolutionary paths for the male and female lineages, all revealed by having that de novo genome sequence that we need that resolves the sex chromosomes. Just say briefly something about long-read transcriptomes. There's a number of, I think, really important applications in plants. We've shown that you really need to go after the long reads, even with long-read sequencing, that most of the transcriptomes in the literature are very deficient in the longer read versions. You see probably only half the transcripts, and they're the shorter ones that you're actually in the published genomes. You really do have to pay attention to that. Particularly valuable, of course, are polyploid transcriptomes, which are incredibly complex, but we do need to understand which genomes are being expressed. And sugarcane is where we've seen a great opportunity. Here we've painted the chromosomes of the two progenitors, A and B, and the hybrid modern sugarcane is a hybrid between an octoploid and a tetraploid, which gives you a 12-ploid genome with some rearrangements between the chromosomes. So understanding what we've done there has been made possible by looking at a system where we could actually look at the transcriptome of the true progenitors, and we actually had a hybrid that was produced. There was one case where we knew the progenitor genotypes from which the hybrid sugarcane was derived. So we can determine, because of the ability to distinguish the transcripts coming from the two subgenomes in the hybrid, which of the subgenomes was contributing each of the transcripts. And that's allowing us to think about how we can reconstruct sugarcane by combining different combinations of chromosomes in this 150 plus chromosome monster to get a more desirable combination of stress tolerance and sugar content and other attributes by mixing and matching the chromosomes, by being able to trace each transcript back to the subgenome. And that's been a very, really useful application of long-read sequencing of transcripts. So we're also now moving to do a lot more spatial transcriptomics at organ-specific, tissue-specific, and cell-specific levels. And we've recently done a major study in dragon fruit. I'm particularly interested to talk to anyone who's doing long-read spatial work. That's an area where I think we need to go, particularly in polyploids, to understand at the cell level which of the versions of the gene are being expressed and which transcripts we're dealing with. We're also using HiFi guided assembly to recalibrate the genome sizes of plants. If we look in the literature, I think that most of the flow cytometry derived genome sizes are fairly wrong. When we assemble the genome, we get a very different result. In the past, it used to be a bigger genome. Now it tends to be more a smaller genome. But quite often the discrepancy is 30 or 40% between the genome size and the flow cytometry. And now we're starting to bring that together. So we're studying in some systems the evolution of genome size, where we've got big genome size differences across the group. But we're now bringing, by becoming more accurate in our flow cytometry and with HiFi genomes, to a point where we're getting almost the same result, which says that maybe we've got the right result at last. There's a couple of good examples of that, where we've got species where we've got exactly the same now in quite big genomes, but that's taken quite a lot of optimization of both genome assembly and flow cytometry. I just wanted to say something about the broader issues around other non-food uses of plants, where we're using HiFi genomes now to breed medicinal plants for alkaloid content, and we can put the pathways together by having all of the genomes at a high level. I think the main point I wanted to make in this regard is that we're looking at using plants to replace oil. So we're currently using fossil carbon, and we've got problems with that. The way to solve that is to use plants as a renewable source of carbon. And this is for things that we can't replace by solar and wind. This is not for energy. This is not for biofuels. This is about plastics. This is about carpets. This is about clothing. They're all made from oil, and we need to make them from renewable sources of carbon, and that has to be a plant, and so we have to reinvent plants. We have to know a lot about their genomes to do that, and a lot of what we're doing is being driven by our ability to have de novo genome sequencing. And so it's about positioning plants not for the old idea of producing ethanol for cars, because that's being electrified, but it's about producing carbon for plastics, fabrics, building materials, and still for aviation fuel. We can fly short distances on electric planes, but we actually need plants if we're going to fly from the US to a meeting here about this technology. And so I'll just end by thanking all my co-workers in the work I've been talking about. And the work is continuing on the de novo domestication and crop work in our Centre of Excellence for Plant Success in agriculture and nature, and the work on using plants to replace fossil carbon will be in a new centre just starting on engineering plants to replace fossil carbon. Thank you.
Thank you very much.